Great. We just went over some basic understanding of how to read a pedigree, so we could interpret, circles as females, squares as males. We have these symbols now that we can use to help us interpret future um, genetics problems. Because there are more problems, we're going to go into a little bit of more advanced Mendelian genetics work. It's not just simple dominance recessiveness anymore. Now we're getting into much more sort of the exceptions to the rules, dealing with things like what happens when two alleles are dominant at the same time, or when neither overwhelms the other and you have incomplete dominance, or when there are multiple alleles involved, or even when the genes are linked together. Mendelian inheritance, what Gregor Mendel was describing, talks about how chromosomes assort completely and independently of each other. A different trait found on a different chromosome, each displaying full dominance and recessiveness, like tall plants and short plants. We got that. But the thing is, that's not the way the world works all the time. So we need to discuss different types of alleles. And when we're describing alleles, we can not just describe them as dominant and recessive, we just can describe them as wild type or mutant. The wild type allele is the one that you see most often in nature. It's the one that's most common. The recessive allele, the mutant allele, is the one that's not seen as much. It's just not around. For instance, here on the bottom left of the screen, you see a box turtle. This is currently the state reptile of Virginia. Who knows, when you see this, maybe there'll be a coup and we'll have like the state reptiles, the copperhead or something. But for now, the state reptile is the turtle, the common box turtle. It is everywhere. I've had to save many of them from the road, according to my wife. I had a really stupid box turtle. I didn't have him. There's a stupid box turtle lived in on my street. I was on this big hill and started raining, like torrential rain. And this box turtle got caught in the rain and started spinning, sliding all the way down the hill toward the sewer grate. So I grabbed the turtle. I was like, cool, save you, turtle. I put the turtle back onto the road. I ran out into the pouring rain because my wife made me. Put it on the sidewalk. Turtle decides to just jump right back into the stream of water and go. So then I had to run back out to the turtle, grab it, and take it and put it by a stream near the house that was also flooding. So, long story short, turtle's fine. It was a common box turtle. They're everywhere. On the right side of the screen, you see the Teenage Mutant Ninja Turtles, or at least a rendition of them. There are only 4 of them. They live in New York. They are by far less common than the wild type box turtle. Now, hypothetically speaking, if there was some sort of, I don't know, turtle Armageddon, and all the box turtles got wiped out, and the mutant turtles proliferated, and what you saw everywhere were mutant turtles, Teenage Mutant Ninja Turtles, those would become the wild type, and the mutant allele would be the box turtle. It all depends on their relative frequency in nature. I want to remind you at this stage about the simple molecular basis for these traits. Remember, when we're looking at traits, whether they are colors or whatever, it's all based on genes. Genes are polypeptide encoding regions of DNA that are found on chromosomes. And each chromosome has a certain set of genes on it, and there's a homologous chromosome in diploid organisms that might have a similar gene but with slight differences that we call an allele. These genes produce a protein. So the DNA produces a piece of RNA. That RNA will go onto a ribosome to translate into a polypeptide that then folds according to its hierarchy of structure to give it a specific form, which gives it a function. Now, if the coding in the DNA is changed, it may change the amino acid sequence, which will change the form of the enzyme or the form of the protein, which changes its function. So you may have functional proteins, non-functional proteins, or some in between. Because of that, if you have 2 different alleles, one of the alleles may function really well and may produce a protein product. The other allele might be broken and not produce anything. So you might have half the amount of protein produced. Now, in simple dominant recessive relationships, half the amount of protein produced still gives you exactly what you need. especially when it comes to enzymes. It's you may need to have two copies of a working allele so you can have the right amount of enzyme. Because remember, enzymes work best in higher concentrations. So half the amount of enzyme might make that reaction rate go slower, and you end up having some trait not coming off as it would have if you had two functioning alleles. So we're going to go to several different scenarios in which there are differences in the amount of protein product produced. One of which is when we deal with sex chromosomes. Now, sex chromosomes, as you remember, are non-homologous. That is, what's on the X chromosome is not necessarily what's on the Y chromosome. The Y chromosome is a bunch of leftover genetic crap. There are very few genes on it. It's much, much smaller compared to that X chromosome. These sex chromosomes link directly to the sex of the individual. Remember, XX is female, XY is male in humans. Now, in humans, XX is female, XY is male, but that's not necessarily the case for all the animals everywhere. In bees, unfertilized eggs become male drones. In certain reptiles and fish, sex is actually controlled by environmental conditions. In clownfish, they're proto-androgynous hermaphrodites. They will go from being male to being female if they're placed into a single-sex environment. Alligators and crocodiles, their sex is going to be determined by the temperature at which their eggs incubate. So it's not even there's sex chromosomes don't play into what their sex is. In humans, when we look at the X and Y chromosome, the Y chromosome is much smaller. It has about 10 times fewer genes on it than the X chromosome. That means that if there's going to be an issue, it's going to likely be an issue with an allele on the X chromosome because the Y chromosome doesn't have any allele to compensate for it. So sex-linked genes refers to genes that are found on one chromosome but not the other. There are some genes on that Y chromosome that are present on the Y but not on the X. There are a lot of genes on the X that are present on the X but not the Y. Usually when we talk about sex-linked genes, we're talking about X-linked genes. Human males are termed hemizygous. They're not homozygous because they don't have two of the same allele. They're not heterozygous because they don't have two different alleles. They're missing an allele for a lot of the genes that are on the X chromosome, so they can't be homologous in those areas. That leads to that special term of hemizygous. They only have one allele. They don't have two alleles for some genes. Females, women, for many X-linked genes, might be termed carriers if they have a defective allele, but they are not displaying that defective allele. They're not showing that defective allele. So they've got the defective allele, they've got an X, let's say X big A, normal, and an X little a, abnormal. They are phenotypically normal, but they are carrying the allele. And as you can see in this pedigree here, which we just learned how to read in the last lecture, you've got a lot of these females are showing as carriers, whereas the males are showing as fully affected. When you see a pedigree that has primarily males that are affected, start thinking that it might be a sex-linked condition or an X-linked condition. So here's a problem, a sex-linked problem I want to walk you through. A man with hemophilia, which is a recessive sex-linked condition, has a daughter with a normal phenotype. She marries a man who's normal for the trait. What's the probability that a daughter of this mating will be hemophiliac? A son? If they have 4 sons, will be the probability that all 4 will be born with hemophilia. All right, so let's walk through this step by step, just like we did our other problems. So a man with hemophilia, a recessive sex-linked condition. So he has hemophilia. We have to ask ourselves right away. First what we have to do is define our alleles. That's always the first step, define the alleles. X big A is normal. X little a means you have hemophilia. You have to have, if you have an X big A, you are normal. So, we have a man with hemophilia. He's a man, which means we automatically know that he has an X and a Y chromosome. He has hemophilia. That means he has an X little a and a Y chromosome. All right, so that's the first thing we know. The man has an X little a and a Y. He's a man, so he's XY, and he has hemophilia. He has a daughter, and the daughter has a normal phenotype. Because she has a normal phenotype, she has to have at least one X big A. And because she's this man's daughter, the only thing he can pass to her is the X little a. If he passed a Y, it would be a son. So she's got an X big A because she's normal, and because she's a girl, she gets that X little a from her dad. We don't know what mom's like. Nobody cares what mom's like anyway. And we get X big A, X little a. All right, so we've got this daughter. The daughter goes and finds a boy. That boy, let's see, what is he? She marries a man who's normal. If he is normal, it means he has to have an X big A. Can he have an X little a? No, because then he would be a woman. And they couldn't produce offspring. So we know he's a man. He's normal, so he's got an X big A and a Y. And that Y is there because he's a man. All right, they're going to mate. So we've got the parent genotypes. X big A, X little a, X big A, Y. We then take their gametes, we then take their diploid state and we divide them into gametes. So the woman's gametes are X big A, X little a. The man's gametes are X big A and Y. We create a Punnett square just like we normally would. And we end up with the woman across the top, X big A, X little a. The man's gametes going down the side, X big A, Y. And the question then asks, what's the probability that a daughter of this mating will be hemophiliac? Well, it's asking only about a daughter. They have a girl. We know it's a girl. What's the chance that she'll have hemophilia? And the answer is 0. Both X big A, both of the daughters on that top row have X big A. All right, what are the chances that they have a son and that son will have hemophilia? And the answer is half, 50-50. One of the sons has X big AY, the other son has X little ay. Okay, a little bit harder. If the couple has 4 sons, these are independent statistical events, what's the probability that all 4 will be born with hemophilia? All right, so they have 4 sons. So we know it's a 50-50 chance. They have 1 son. It's 50-50, so 1/2. Another son. To get a combined analysis, it's 1/2 times 1/2. So there's a 1 out of 4 chance that they will have all 4 sons are born with hemophilia. If the couple, let's see, if the couple has 4 sons, if the couple, so now 3 sons, 1/2. So 1/2 times 1/2 times 1/2 times 1/2 is 1 over 1 over 32. 1 over 16. 2 times 2 times 2 times 2. 1 over 16. That's the probability that all 4 will be born with hemophilia. Not all traits that people think of as sex-linked are actually sex-linked. Male pattern baldness is one example. Male pattern baldness is sex-influenced. It is something that is linked to the X chromosome. That is true. But it only shows up in males because of the hormones that are produced by males. High levels of testosterone, low levels of estrogen. Women who have this same genotype, X little a, for example, they don't show male pattern baldness when they're younger because their testosterone levels are low. But as they get older, they may start to see male pattern baldness creeping in. So you'll start to see some older women with displaying male pattern baldness because they are both, they have the recessive alleles, and they're lacking the hormones that were masking it when they were younger. So as you can see from this pedigree, females are typically carriers of male pattern baldness. And if a male inherits even a single X chromosome that's affected, he will have the condition and display it early in life while he's producing testosterone. Another idea, but separate from sex linkage, is incomplete dominance. With incomplete dominance, you're going to see the heterozygote displaying an intermediate phenotype. Previously, you might have seen red and white, with red as dominant to white. And you would get, if you cross a homozygous red plant to a homozygous white plant, you get all red plants. But instead, this time, you're getting pink plants. Pink is an intermediate phenotype between red and white. It'd be as if you'd make two horses together, a black horse and a white horse, and you get a gray horse. These alleles are incompletely dominant. You get a mixed phenotype. In humans, there's a condition called phenylketonuria, PKU. And PKU is a incompletely dominant condition. It's caused by an enzyme whose job is to break down phenylalanine. When it doesn't work, when the enzyme is completely broken, phenylalanine levels build up in the body, and they lead to toxicity that could cause mental retardation, decreased body growth, might cause microcephaly, all sorts of developmental abnormalities. When they're looking at these levels of PKU, what doctors have found is that if a person is heterozygous, they have 4 times the amount of phenylalanine in their blood as a normal person would. But if they are homozygous recessive, they're going to have a grand total of 28 times more phenylalanine in the blood than a normal person. So you see this is an intermediate or an in-between type. It goes normal blood levels, 4 times too much phenylalanine, and then 28 times too much phenylalanine. Intermediate. We can do a simple intermediate inheritance problem, or incomplete dominance problem, by thinking about those red and white flowers we just talked about. We're going to say in flowers, red fruit is incompletely dominant to white. I'm sorry, red flowers are incompletely dominant to white. Heterozygotes create an intermediate color that's pink. A plant that's homozygous for red flowers is crossed with a plant that has white flowers. What would be the genotypes and phenotypes of the P, F1, and F2 generations? So tell me all about these plants. All right, so we start off. We got to, just like always, we start off labeling our alleles. Big R is red, little r white, big R, little r is pink. You can use whatever letters you want. I don't care how you do it. We've got our alleles labeled. Now we need to figure out our parents, our P generation. Well, we're told that we have a red fruit plant and a white plant. The only way they can be red is by having 2 red alleles, so big R, big R. And the only way they can be white is by having 2 recessive alleles, little r, little r. So we got 2 parents, big R, and little r, little r. If we had a heterozygote, big R, little r, it would be pink, and we'd know it. We then create gametes from that. Big R makes a big R and a big R. Little r makes a little r and a little r. Create our Punnett square, and we see all heterozygotes. So based on that, we have a 1-to-0 ratio of heterozygotes, and every single heterozygote's big R, little r, which means they are all pink. Great. There's our F1 generation. Now we talk about our F2. For F2, we cross the siblings together. Big R, little r cross to big R, little r. We get our gametes. Big R, little r forms a big R and a little r. Big R, little r forms a big R and a little r. Then we create our Punnett square. And we can see that same ratio we saw when we crossed them in previous lectures, that 1 to 2 to 1 ratio, 1 to 2 to 1 genetic ratio. So 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. But the phenotypic ratio looks very different. Because now you have, instead of a 3-to-1 ratio with simple Mendelian dominance, we have a 1 red to 2 pink to 1 white ratio. Another idea of an imperfect world is multiple allelic inheritance. In a natural population, you're going to usually have multiple alleles of a certain trait, not just 2 alleles, but multiple alleles. And these alleles may have different dominance and recessive relationships to each other. There's a lot of variation in a population, which is good because otherwise we die out. That's the way evolution works. In mice, you might have 3 colors: black, yellow, and agouti. And agouti is sort of a combination of colors. And this inheritance is due to multiple alleles. I'm going to ask you what's the phenotype of the offspring from a cross between a heterozygous yellow allele and a heterozygous yellow mouse and a heterozygous black mouse, both of which had an agouti parent. Black is dominant to yellow, is dominant to agouti. Sorry, I just love saying agouti. So let's work it all out. Here's our problem again. We need to figure out step by step what we do. And the first step, as always, is label your alleles. In this case, I have H for hair, capital B is black, capital Y is yellow, little h is agouti. Figure out our parents' genotypes. All right, so take a look at the parents here. We know it is a heterozygous black and a heterozygous yellow, each from an agouti parent. So we know that it's black, and its parent was agouti, which means it has to have a little h. They are heterozygous. Same with the other one. It's yellow with an agouti parent. So you know that it has to have a little h, and it has to have a y because it was displaying yellow. We come up with the gametes like normal. We take the first one, you take HB and H and split them. So you have HB and the gamete H. You split the other parent, you have HYH is going to split into HY and H, and you put them on different axes of the Punnett square. And you end up with this particular Punnett square where you've got HBHY, HYH, HBH, and little h. So we ask ourselves, what are the ratios? Our genotypic ratio is 1 to 1 to 1. Each of these genotypes is distinct and different. Our phenotypic ratio, on the other hand, is 2 blacks, so that first column, and then 1 yellow and 1 agouti. After I talk about multiple allelic inheritance, usually I like to talk about codominance. Codominance is where you can see both traits at the exact same time. So when a single individual expresses multiple alleles simultaneously, if the image on the screen, it is not a pink flower, an intermediate inheritance, it is a red and white. We have a red flower crossed to white flower, we end up with a red and white flower. It would be like if you took a white horse and a black horse, you crossed them together and get a zebra. That is not the way it works. That's not the way zebras come about, but it demonstrates the idea. You're seeing 2 traits simultaneously expressed. To give you a sample of this, in flowers, red color is dominant to white flowers. I'm sorry, codominant with white flowers. A plant that's homozygous for reds crossed with a plant that has white flowers. What would be the genotypes and phenotypes of the P, F1, and F2? So this is a very, very similar problem to the ones we just did. Step 1, label your alleles. In this case, I've got big R's red, little r's white. You can use whatever labels you feel like using. You can use R's and W's. You can use, I don't know, R and then R prime. Whatever you want to use, whatever you're comfortable with. You just have to keep them separate. We look at our parent genotypes and we know that the red plant has to be homozygous for the red allele. The white plant has to be homozygous for the white allele. So one parent's big R, one parent's little r. We create the gametes from that, and we get big R and big R, little r and little r, And we can put that into our Punnett square to see that, once again, the entire F1 generation is heterozygous. If they're heterozygous with a codominant alleles, though, they're not all red. In this case, they are red and white. They're displaying both red and white colors simultaneously. So the genotypic ratio is they're all heterozygous. The phenotypic ratio is they are all red and white. Okay, so we take our red and white flower, we cross them to each other. Now our parent genotypes are big R, little r. Our gametes, of course, are big R and little r for both parents. We create the Punnett square. And you see that you got that 1 to 2 to 1 ratio again. It's magic, right? It happens a lot. Well, for this, you see the genotypic ratio is 1 to 2 to 1, and our phenotypic ratio matches it. 1 red to 2 that are both red and white to 1 that is white. The reason I talk about codominance and multiple allelic inheritance together is because it leads to ABO blood typing. This is human blood typing. The way human blood typing works is you could be type A, type B, type AB, or type O. And it's all based on proteins that are on the outside of the blood cell. If you have type A blood, in this case you might have, I don't know, what are those, circles as a protein on the outside of the cell. Your immune system is looking for circles. It only likes circles. If there's anything that's not a circle, it will kill it. It will lead to an extreme reaction. For blood type B in this case, you might have diamonds that your cell membrane has. Those diamonds, your body then is looking for diamonds. If it finds circles, it will attack the blood that has circles. If you're blood type AB, see, multiple alleles, right? You got an allele A, an allele B, and an allele little I. They're codominant, so AB is showing both diamonds and squares. This person's body is looking for diamonds. It's looking for circles. It's looking for anything. Any type of blood you put into it, it will not react to because it expects to see the antigens, the proteins that are on the outside of A blood or the antigens that are on the outside of B blood. And then finally, type O, they have absolutely none of these proteins on their membrane, which means that their body, if they recognize that there are any proteins at all on any of the blood cells, it will attack and kill them. So blood type AB can accept a blood transfusion from any other group. Blood type O can donate blood to any other group. Blood type A can only accept from group A or from group O. Blood type B could accept from group B or group O. The alleles that you're looking at are for type A, type B, and type O. Type O is fully recessive, so we label that as little I. Little I is type O. A and B are codominant to each other. So to be blood type A, you could be capital IA, or you could be capital IA, little I. The only way you could be blood type AB is by having alleles capital IA, capital IB. So let's move into how we can use this to predict, to detect paternity, because this used to be used for paternity testing for the longest time. The downside is it can't tell you who the father exactly is, but it can tell you exactly who the father isn't. In humans, the blood groups are produced by the various combinations of the three alleles, IA, IB, and I. Suppose the daughters of blood type A and the mothers of type O. What type or types may the father belong to? All right, so we got the question here. I got a big open space to work it out. First things first, same as always, write down your alleles. When in doubt, write down your alleles. Next, we've got to figure out, suppose the daughter's of blood type A and the mother's blood type O. So let's talk about the daughter. The daughter can be IAIA or IAI. We don't know. She's blood type A. Who knows what she is? She could be either homozygous or heterozygous. The mother is blood type O. So we know for sure that she is little I, little I. That's the only way it can be blood type O. So let's start off. We've got the mom. Because the mom has little I, we know that the daughter has to have a little I. So there's only one possible genotype the daughter could have, IA little I, which is right there. Now we got to figure out the father. Well, if the daughter's genotype is IA little I, she has to get the IA from her dad. So we know the dad has to have an IA. We don't know anything else about him, just that he has to have an IA. So if we look at the possibilities, any of these that have an IA in them could be the father. And that really just comes down to these two groups. The father could be type A or type AB. If the man that the person they were trying to figure out was the father was type B or type O, they're out of the running. Try another one. In this case, we have a daughter's blood type B and the mother's type AB. A little bit harder. So we got our first step, which is to label all of our alleles. Our next step is to record what the daughter is. The daughter is type B, which means she could be IB, or IB little I. The mother is AB for sure. Now, that's useful. We know the mom's AB, but that doesn't do much as far as figuring out what the daughter is. The daughter could get the B from mom. I mean, obviously the daughter did get the B from mom, but she could get a B from dad or an I from dad. So we don't know if the daughter's IB or IB little I. No idea, so we have to account for all of that. Then we ask ourselves, what is the father? The father must have either an IB or little I. We don't know. So looking at our possibilities, we scan through here to see what are options that have either an IB or little I, and it turns out it's every single one of them. Which means with this combination of mother and daughter, you cannot rule out any man on the planet from being the daughter's husband. I'm sorry, the daughter's father. Yeah, and this is exactly why they don't use ABO blood typing to determine paternity anymore. Use genetic fingerprinting, which is like 99.8% effective. Last paternity test, real story. We have a daughter is of blood type AB and the father is type O. What type or types may the mother belong to? A student in my class previously came to me, and they said, Dr. Pantasis, I am type AB. My dad is type O. There's no way ABO blood typing works this way. So we went through it. We labeled our alleles. The daughter we knew was IB for sure. The father we knew was type O, little I, and we created our graph. Well, what's the mom like? We don't know yet. The daughter's AB, the father's little I. There's a reason this is impossible. It's an issue. That daughter has to get an allele from dad. There's no little I in the daughter's blood type there. That means that this is not logically possible. So what does that tell me? Okay, well, what it told the kid, because all I told this student was, hey, so blood typing does work this way, and doing this pedigree, you can see that none of those blood types work. So maybe you should go home and talk to your parents. And it was at that very moment, not that moment, but the next class meeting they came in and they told me, yeah, it turns out I was adopted. So luckily there's no infidelity or anything, which is often how something like this might happen. The kid found out they were adopted because of their general biology class. So you too can learn things about yourself in college. Another concept I want to cover is gene linkage. Genes that are on the same chromosome do not independently assort. They are linked together. They're held together by basically a chain. So if you have two genes, A and B, and they are on the same chromosome, they're going to travel together. So you're not going to see complete independent assortment like you do with the other dihybrid crosses we've done so far. They tend to come together, they're inherited more often than not with each other. Now, not all the time. Sometimes they don't travel with each other, and that occurs due to when crossing over happens in prophase of meiosis. It was using linked genes that we were able to initially map out genomes by saying, cool, these genes travel together more than 50% of the time. And because of that, they're consistently traveling together more than 50% of the time, we could actually map out distances between them. Ones that travel together close to 50% of the time tend to be close to 50% of the time tend to be farther apart. And those that travel together more than 50% of the time, much more than 50% of the time, tend to be close together. So there we go. A lot of things going on in this lecture. Incomplete dominance, sex-influenced traits, codominance. We talked a little bit more about Mendelian inheritance and gene linkage. These are still basic Mendelian traits. They still have dominant recessive relationships to each other that can be calculated. This is probably the end of the problems that I ask you to calculate out. From here going forward, it's just concepts. But in each of these, incomplete dominance, sex-influenced traits, codominance, and Mendelian inheritance, likely you're going to have practice problems on these. Speaking of practice problems, here are some you can work on. We covered a lot. Mendelian inheritance is just simple dominant recessive relationships. We talked about how a gene will encode for a protein, and sometimes that protein is fully functional when there's only one copy. Sometimes it's partially functional, and sometimes it is expressed simultaneously with another allele. We talked about sex-linked genes that are on the X or Y chromosome. We talked about incomplete dominance, in which you get an intermediate phenotype when you cross two pure breeding strains. We talked about codominance, where you get a phenotype that shows both alleles at the exact same time in the organism. We talked about multiple allelic inheritance, with many alleles in the population with different dominant recessive relationships to each other. And we talked about gene linkage, where two genes can be attached to the same chromosome and get transmitted together. Whew, yeah, it's getting rough, huh? These are the content review questions to really focus your studies. And speaking of rough, now we're going to move into definite non-Mendelian concepts that are a little bit more sort of way beyond Mendel and more advanced modern genetics concepts in the next lecture.